Welcome everyone to today's webinar. My name is Christiaan and I will talk a little bit about fluorescent lifetime imaging microscopy in electrochemical flow reactors. But let's start a little bit with who I am. So my name is Christiaan Schinkel. I have a background in physics and I started working at the Delft University of Technology in 2015. I'm a research and education associate or a technician. And over the years, I've specialized in optical laser-based techniques, such as particle image velocimetry, FLIM, and high-speed imaging. Today, however, we'll only talk about FLIM. And I prefer to stay at a conceptual level and reduce the amount of mathematics. So I'm working in the David Vermaas group. We work on electrochemical flow systems, and that means we're interested in the conversion of renewable electricity into chemical products and fuels. So the research focuses on electrochemical conversions, and in particular to the mass transport on a microscopic level. So in 2020, we were granted an ERC to enforce breakthroughs on transport limitations on in electrochemical reactors. And that's a mouthful, but let's try to make this well, a little smaller. So over here I have a simplified model of a reactor where we try to do CO2 electrolysis. So we have a current running from one side to the other, and we're converting carbon dioxide to carbon monoxide. And carbon monoxide is a feedstock chemical for the chemical industry. And of course, if you build such a reactor, you want to have a high reaction rate so that you can convert a lot with the same reactor. But of course, at some point you start putting more current in, but the carbon dioxide that is dissolved in your liquid will all be used up. And if there's no carbon dioxide anymore, there's a competing reaction where we produce hydrogen, which is not what we want. But the question is, what is now actually happening close to the surface of this electrode? Is it actually CO2 depletion? How close is that? Can we do some tricks to enforce convection? So we need a technique to measure close to this surface. And this is one example, of course. So you could think of fluorescence intensity imaging. So regular fluorescence imaging. But then you're always dependent on the dye concentration or the illumination of your sample. And then you don't always know whether the structures or phenomena you see are because of the local pH or because of the concentration, for instance. And if you do FLIM, you are completely independent of that. If you have a low dye concentration, it just means you need to measure for longer. And if and this also applies if you have a bad illumination in a certain region. Yeah, so FLIM enables us to quantify pH gradients. Yeah, so the plan is we build a custom reactor where we have optical access so that we can measure the local pH profile. Then once we have this tool, we can check some process parameters. What happens if we change the current density, the flow rate or the concentration, for instance. So I was allowed to go shopping and this is what I bought. This is the toggle. It's a special camera from Lambert. And unfortunately, it's not available anymore, but they have a nice successor. This camera is able to take instant FLIM images at a reasonable resolution with a nice sensitivity. And what makes this camera so special? Well, let's start by explaining how a regular CCT camera works. So the idea is that we have a photon falling in, hitting the photosensitive layer, liberating an electron, and the electron goes to a register or an electron bucket. You do this over a certain integration time until you've caught enough light, and then you count how many electrons are in your register. The toggle has an extra feature in there, and it looks a little bit like this. So the photon still comes in on the top, it liberates an electron, and then on a pixel level, they have an electric field where they move the electrons to one end or the other. So we're basically having two registers, and we're taking two images at the same time. So if we go back to the signal that we're trying to measure, so over here you see in blue the excitation source, and then the fluorescence has a phase shift, a delay, and also the amplitude is weakened a little bit. And these are both measures for the lifetime of the fluorophore that you're measuring. So we use the toggle, and basically we're exposing the left register in the yellow rectangle here, and we're using the right register in the red rectangle over here. And well, that would already supply us with a FLIM image, but if it's not sufficiently precise, we can of course take another image with a phase shift. And yeah, you can do that at different phase shifts, making sure that you're better capable of measuring this fluorescence sine wave, which allows us to calculate the lifetime. And you're doing this per pixel. So, well, this is the principle. In the lab, it looks a little bit like this. It's a little bit outdated image, but all the important components are in. The laser is behind the microscope, going here with a fiber probe towards this black unit. The black unit here is it's a spinning disc confocal unit. It's, well, for this story, it's not so important to explain what it does. The light goes here. It comes out of the objective over here. We put our sample or reactor or whatever we want to look at. Then the fluorescence goes back through the objective, goes back to the unit. Over here we have some filters and a dichroic, and then the fluorescence gets back to the toggle. So in the schematic it looks a bit like this. So we illuminate through the objective, we have our setup, the fluorescence comes back, we move it to the toggle, and we can take our FLIM image. So, all set, ready to go? Well, almost. I guess there's a lot of biologists in the audience, and I wonder if they ever worked with living cells that like to live at pH 10 or pH 12. I don't think there are any living cells that survive that. And that also means that the industry doesn't have suitable fluorophores for us. So yeah, there were no commercially available dyes, but lucky for us, we have a few nutty professors in our department, and one of them made us some dyes himself that are sensitive in the high pH range. So for that, I'd like to show this little graph. This is a demonstration of the 5 dyes that were described in this paper that are sensitive to high pHs. Well, we have had plenty of students now that were happy to use these to do pH probing at high pHs. So, of course, I would like to show some fancy results, but for that I give the floor to Laura. And I would like to thank you for your attention.
Thank you, Christiaan. So, Laura, now the floor is yours.
Yeah. I'm gonna share my screen. Yeah. Yeah. So thank you, Christiaan and Debbie, for the introduction. I'm Laura Forero. I'm a master's student in chemical engineering inside the electrochemical fluid systems group. My background is also in chemical engineering and industrial engineering. And in my project, I'm working on pH imaging using FLIM in electrochemical flow systems. But the system I'm interested in is anion exchange membrane water electrolysis. And this combines the benefits of technologies that have been widely used in industry for water electrolysis. And some of them is that it is typically operated at alkaline conditions, and this allows to use materials and metals that are stable in these conditions and are cheaper and earth abundant. So we have the nickel and its alloys as some of them. So, but why I studied the local pH in this type of systems? So here I have a very basic representation of how the anion exchange membrane water electrolysis works. So in the middle, we have an anion exchange membrane that allows the transport of anions, in this case, the transport of hydroxide ions. And on one side, we have the production of hydrogen. So in the cathode side, we have the production of it, so consuming water to produce hydrogen and hydroxide ions. On the other side, we have the consumption of these hydroxide ions and the production of oxygen and water. So we expect that on the cathode side, the pH goes higher because we are producing OH minus. While on the other side, we expect that locally we will have a consumption of OH minus and therefore the pH will be lower. But this can affect the kinetics and the stability of nickel. So we want to explore further how does it look the local pH in our cell. In this sense, my project is titled Local pH Mapping for Nickel-Based Anode in anion exchange membrane water electrolysis. And usually we see reactors as a black box where we change the input variables and then we study the output variables. But there is where technologies as FLIM allows us to understand what's happening inside the reactor. So with FLIM, we will be able to see the pH gradient inside the cell, and this will give us insights of things like the boundary layer development, mass transfer phenomena that is happening inside. And for example, we can compare the pH in the surface versus the pH in the bulk, which will give us some insights that will tell us about if the nickel will be stable or not in these conditions, and if the conditions are harsh for it. For that, we designed a cell that is the one that is shown in the picture. It has two considerations. One type of them is to be representative of the system we want to study, and the other ones were considerations to make it suitable to use FLIM. So for FLIM, we wanted it to be transparent, and also we wanted to avoid gaskets because in previous studies we saw that some of the materials used for them are autofluorescent. So here I have in the middle my anion exchange membrane, then I have the two electrodes, and then we have these flow fields that are made on PMMA, and one of them is optically transparent, so it was diamond milled to make it transparent enough to use FLIM. So this is my cell, and then the setup is something like this, where we wanted to study the oxygen evolution reaction. So the anode side of the cell, where we expect the pH to drop. So with FLIM, we use it, and the image that we got from it will look something like this. So I'm going to show the results, but first I wanted to say that on the left side, we will see the nickel felt that is our porous transport layer, but it also works as the catalyst. And then we will see also the flow, so the flow channel next to it. But what we are interested is in the pH map, but what we get from the toggle camera is a lifetime map. So what we have to do is to translate that map into the pH map. And to do that, we use the calibration curve of the dye. In my case, I used mainly what we call the alpha dye. That is one of the dyes that Christiaan already mentioned before. That is the one here in the bottom. So we see that this dye is sensitive in a pH range between 9 and 13. And from them, we can get a pH map. So how does it look? Here I have a result that is how does it change in time for when I operate the cell with 100 milliamperes per centimeter square and a flow rate of 0.1 milliliters per minute, pumping electrolyte to both sides of the cell. What we can see is that in the first two images, we have a very homogeneous pH gradient inside, so we don't have a gradient inside. But then after applying current, so I started applying current at time 0, we see that in the next seconds, so 10 seconds later and 20 seconds later, we see how a gradient is formed in the interface between the nickel felt and the electrolyte. And we see how that gradient increases in time, but then we see that it is depleted. So our hypothesis here is that it is because of mixing happening inside, and more particularly because of bubble-induced mixing. And we can also see other things. So we see, for example, here that there is a stagnant bubble in the porous transport layer. Here, for example, there was a bubble that was moving through the whole channel, so it eliminated the profile and we see something more darker. But what we see in time at the end is that there was not more a boundary layer or a change in the gradient very localized, but more like the pH change in the bulk also. So we see that the last images are more yellowish than the first ones that are more like orange. Maybe that was not very clear, but we can also translate these pH maps into concentration profiles. And what I did here is that I averaged through the y-axis and got these pH profiles, but that then I translated into concentration profiles. And here I have four different images that correspond to different current densities. So in the first one, I have a very low current density, and in the last one, I have a higher current density. And what we see here, for example, or something that FLIM allows us to study, is that in the first one, we see how the boundary layer, that is the zone where the concentration changes, increases. So initially, it was smaller, and then it goes bigger. Yeah, so we see that for a very low current density, but then we see that with higher current densities, that happened initially, but then it got back. So in the yellow one, we see that it gets back to similar to the initial profile. And this is because there was mixing and the boundary layer was depleted again. But what we see at high current densities is that instead of developing a boundary layers, so growing this distance in this decay, is more like the bulk pH go down. And this is something that we see for the last two. And we can also see this in the steady state. So we see that, for example, in the first image, that was the low current density where the boundary layer grows, we see a boundary layer. So we see that there is a concentration profile and a gradient profile between the interface between the nickel felt and the electrolyte versus the bulk pH. Whereas for the other ones, we see more like a change, an homogeneous change in the pH. So we see that the images are more yellowish. And this is an example of how are we using FLIM to understand what's happening inside electrochemical flow systems. Here I wanted to show a nice video, and First, I'm going to explain what it is. So in this case, we wanted to understand further what's happening inside the nickel field. So not only what's happening here in the interface, that is what I was showing you before, but I did a hole to see what's happening inside, or have an idea of what's happening inside the nickel field. Yeah, so this is how it changes in time. I don't know how to play the video. Huh. Huh. So in time, it changes like this. Yeah, we see that near the surface, it gets more green, then more blue, and that means that the pH went down. So we can understand what's happening in time close to the nickel field and have an idea of what's happening also inside. Oh. And then the takeaways of this part is that I wanted to show you some results of what we get with FLIM in our group. And first, something nice is that we can get a high resolution for mapping local pH dynamics. So in this case, I have a resolution and a spatial resolution of 5 microns for the pixel size, and I was taking frames every 0.5 seconds. Also, it gives us in situ insights of what's happening inside the reactor, which is also allows us to understand further the mass transfer phenomena and, for example, see the boundary layer formation and bubble-induced mixing. And lastly, it's also important to see how the pH changes, and this is something that FLIM allows us to do, because based on the pH, the nickel that is our catalyst and our porous transport layer can change its stability and catalytic activity depending on it. So thank you for your attention, and also hope this example shows us how we are using it in our group.
Okay, thank you so much, Laura. So Geert, this would be your time now. Pass this off into our next presentation.
Okay, thank you. Let me share my screen. Share your screen sharing. Okay, good. So can you see my screen? Is everything working?
Yeah.
Okay, super. Okay, thank you for the invitation and the opportunity. First, a disclaimer. I am a biologist, so you cannot expect any mathematics from me. I do have an interest in cell biology, and to be more concrete, in immunology. So my lab works on immune cells and all organelle trafficking related to that. So you can think of cytokine secretion, antigen uptake, antigen sampling. And I think this is a very fascinating topic, because if you look at the cells, so you see here an electron microscopy image of a human blood-derived macrophage, a type of immune cell, you see that they're actually crowded with organelles. So there are the large thing on the left is the nucleus. But if you look closer, you see many, many organelles. And how do organelles find their destination? How does a molecule reach its destination, traffic from one organelle to the other? That is a question that we are very interested in. And one of the things that we're also interested in, and that is what I think is very nice in the previous talk, is pH. So we use a very similar approach, FLIM-based approach, to measure pH. Different organelles have a different pH. The dysregulation of pH in organelles leads to the mistargeting of proteins, is a cause of disease. However, in this talk, I want to focus on something else, which are the so-called snare proteins. So I will show you how you can use FLIM, so Fluorescence Lifetime Imaging microscopy, to visualize those in cells, and also how to do this dynamically. Now, to remind the biologist and for the non-biologist, so snares are the so-called engines of membrane fusion. So you find them in virtually all organelles except the mitochondria, and they fuse membranes. So you see here an example. In red, you see a trafficking vesicle that has one snare, and then it interacts with cognate snares in the target membrane in black, and they zipper up, forming this tight alpha helical coiled bundle that then overcomes the membrane energy barrier for fusions, for membrane fusion. Now we humans have like 27 different snare proteins or so, and they regulate all cellular transport except mitochondrial fusion. So you see it here. However, the problem is that snares are redundant, so you have multiple snares catalyzing the same pathway. And at the same time, they're also involved in multiple pathways. So if you take, for example, FLIM, and this is a key snare that I'm interested in because it mediates the release of inflammatory cytokines, so the hormones of the immune system. You see that this FLIM is involved in many different organelle fusion steps. So exocytosis, lysoendosomal trafficking, also earlier in the ER trans-Golgi network trafficking pathway. So how do we now know if we make, for example, a mutation, where this affects the role of FLIM? So what we needed and what we developed is a new technique, a FLIM-based technique, to visualize the cellular localization of these snare proteins. Okay, how did we do that? So snare proteins are special because they're so-called, at least most snare proteins are special because they're so-called tail-anchored proteins. And that means that at their very C-terminus, they have a transmembrane region. So you see here the 27 or so snares in the human, and you see there the domain orientation. And on brown, in the far right, you see that most snares have a transmembrane region. This makes them tail-anchored. And that makes them very suitable for FLIM-based approaches. So, okay, so this is FLIM, also still anchored. So why? Because after the snare complex have formed, so you see here 3 snare proteins, in purple FLIM, in yellow syntaxin 4, and then in green is a SNAP 23 from the top of my head. You see that the C-termini are immediately juxtaposed to each other, so next to each other. So if you fuse these to a donor and an acceptor fluorophore for first resonance energy transfer, they are ideally located for this. So the idea is the following. You express in cells cognate snares fused to a donor fluorophore, M-citrin, and an acceptor fluorophore, M-cherry. And then before fusion, you see no first resonance energy transfer. And then after fusion, you get first resonance energy transfer. And this, theoretically, you could measure it radiometrically, so with normal intensity-based microscopy. However, in practice, this is very difficult because you cannot calibrate the system. You always have crosstalk, so meaning that you get the M-cherry signal in the M-citrin, and that your laser that you use to excite M-citrin also excites M-cherry a bit. And it's very difficult to control for this because the protein levels differ between different regions of the cell. So the only way to really do this is fluorescence lifetime imaging microscopy, FLIM. And that works really nice. So you see here a control. So on the top row, you see microscopy images of the donor only. And then you see on the left the simple microscopy, the fluorescence microscopy images. And on the right hand, you see the lifetime imaging microscopy image. In the middle, you then see a co-expression with the acceptor snare. And in the bottom, you see a sort of a positive control where we fuse the donor and the acceptor fluorophore in tandem to syntaxin 3. And what you already beautifully see is that the lifetime in the FLIM image on the right hand is lowest at the plasma membrane. So this is really where FLIM interacts with syntaxin 3 in this particular cell. And what we can now start doing is things like, for example, changing the snare proteins. So on top, you see again syntaxin 3 with FLIM. And on the bottom, you see now that we changed the FLIM for FLIM8. And what you see that for syntaxin 3, FLIM, you see more interaction at the plasma membrane. This interaction is also increased if you activate the cells with the protein or with the molecule called lipopolysaccharide. It's a bacterial toxin, a very potent immune stimulus. Whereas syntaxin 3 and FAMP8, you see more interactions in the endolysosomal compartment. We can also change the syntaxin. So now we move from syntaxin 3 to syntaxin 4. And then you see that the interaction of syntaxin 4 with FAMP3 is also at the plasma membrane. You also increase this with this lipopolysaccharide, this LPS. And syntaxin 4 and FAMP8 do not interact at all. And please notice that you really need FLIM for this. So if you look at the intensity images, there's no way you can tell where the interaction is taking place, right? Because you only see overlap, but it does not necessarily mean interaction. Okay, so that is the technology that we developed. Can we push this a bit further? So can we also use this now to measure dynamic things in cell? And that is what I want to show you next. So I want to show you an adaptation of this where we combine this with the so-called rush system. And before I introduce this to you, I want to explain the use case here. So I was approached by a clinician who identified siblings with a severe genetic disorder called a congenital disorder of glycosylation. And that led to all sort of systemic abnormalities. So you see there's basically not an organ in these infants that is not affected, and they also passed away at very young age. So what we were interested in is why. So they did the deep sequencing, and then they identified a mutation in a snare protein. And this snare protein is called syntaxin 5. And you see here part of the sequence of syntaxin 5. And the mutation was here. So an ATG, that is a starting codon that codes for methionine, as you, at least all biologists in the audience will know. Proteins are translated with initially from an ATG, so that is a start codon coding for methionine. But in these patients, one of the alternative starting codons was mutated into a valine. What did that mean? It meant that only the first ATG could be used. So these individuals only produce the long isoform. And you can nicely show that by Western blot. So please focus on the top blot. You see there that there is a syntaxin 5 long and a short isoform. On the left, there are fibroblasts isolated from control individuals, so healthy individuals. And you see that they make both in a more or less one-to-one stoichiometry. The two patients that you see on the right, or two of the patients that we tested, you see that they completely lack the short isoform because this is a starting codon isoform. All the other snares that we tested, the levels were unaffected, with the exception of BET1L, a cognate snare for syntaxin 5, and I'll come back to that in a minute. So what does syntaxin 5 do? Well, it is very well understood to work in ER, so endoplasmatic reticulum Golgi trafficking. So you see here different sets of cognate snare proteins that mediate the trafficking from the ER to the Golgi and back. However, again, syntaxin 5 here has multiple roles. So it is involved in multiple snare complexes, and it has both anterograde trafficking, so from the ER to the Golgi, and retrograde trafficking from the post-Golgi back to the trans-Golgi, back to the cis-Golgi compartment. So, of course, these patients, even though there's infant mortality and they all passed away within several months after birth, but still the organs formed, so the organelle, the development was of course clearly affected, but not that dramatically affected, right? This mutation was viable. So what we expect at the cellular level is a subtle phenotype. And this is of course very difficult. So how do you find that? So we tried expressing these snares using this FLIM approach, but we did not see a lot. And that is why we then decided to use a trick, and this is what I will explain you now. We combined the FLIM approach with the rush system. So you see the concept here, and this I realize this is a complex slide, so please bear with me. RUSH stands for retention using selective hooks. So what you do, and you see it on the left, is we express a syntaxin protein that is fused to the donor fluorophore, the M-citrin, and we also fuse it to SBP, and that stands for streptavidin binding protein. So streptavidin binding protein. And as the name suggests, it binds to streptavidin. So we co-express this molecule with streptavidin that is fused to an ER retention signal, KDEL. So a hook, retention using selective hooks. So the hook is streptavidin fused to KDEL. And this, what this does is, in absence of biotin, it retains the syntaxin construct into the ER. And what you then do is you add biotin to your cells. The biotin will bind to streptavidin and thereby chase off, so by competition, the snare protein, and it will reach its cellular destination. So on the right, I will play the movie. So what you see here are 3 cells. We start in a condition where there's no biotin, and the syntaxin 5 is in the ER. And now I will play the movie, we'll add biotin, and what you will start seeing is that it moves to the Golgi. It does not move beyond the Golgi because this is a Golgi snare, as I just showed. So there we go. You see that you add biotin, it moves to the Golgi, and it stays there. Okay, so now let's increase the complexity. So what we now do is we express a third protein, and that is the acceptor snare. So in this case, we just show you part of the data. We express it with BET1L fused to the acceptor fluorophore. This is a Golgi snare, so in absence of biotin, syntaxin 5 with M-citrin, so the donor is confined to the ER because it binds to the hook, the KDEL. M-cherry BET1L is mainly at the Golgi, so we expect no first resonance energy transfer. Now we add biotin to the cells. The biotin chases it off, and we start seeing the first resonance energy transfer. Okay, does that work? Yes, it does. So you see here 2 different constructs, the syntaxin 5 short mutant. So this is starting from the first ATG, so the first starting codon, and the second, and the long variant, and this is basically the patient mutations where the second chain has been mutated into a valine. And what you see here is that BET1L is in the Golgi, so this is the Golgi, it's very fragmented Golgi in these cells. Syntaxin is mainly in the ER, so they do not co-localize. You see it here in the merge. You also see the lifetime, that they have a very long lifetime. It's quantified here because they locate in different compartments in the absence of biotin. Now we add biotin, so we chase away the syntaxin to the Golgi. So you see here that both the syntaxin and the BET1L is in the Golgi, and we measure the lifetime. And what you see here is that the short form has a lower lifetime, meaning more interaction with BET1L compared to the long syntaxin form. Thus, what does that mean cellularly? And I'm again not showing you all the data, but just the interpretation. It means that the short form plays more a role in the Golgi, whereas the long form plays more a role early in the ER trafficking. And because the patients only have the long form, you get a wrong distribution of your anterograde versus your retrograde trafficking. And what we could also show is that this then leads to mislocalization of glycosyltransferase enzymes that are normally restricted to certain layers of the Golgi apparatus, and therefore to the glycosylation effect that then in the end affected these patients, so caused the symptoms of these patients. Okay, and that is what I wanted to show you. So I hope I convinced you that FLIM does allow you to visualize snare complexes in cells, and that you can even combine this with the Rush system to visualize snare complexes along the exocytic pathway. And I should also mention that that is why we are very interested in this Lombard camera, and we also have it here now in our lab, because we can now do this dynamically. If you use a laser scanning system, you need to accumulate sufficient pixels per photon. So the speed there is very slow. It takes minutes to record a whole cell. And now you can do it in a second resolution. So we can measure these processes dynamically. Okay, that's what I wanted to show you, not without thanking the people that did the actual work. And thank you for your attention.
Great. Thank you, Geert. That was very informative. So, Johan, do you want to give your little 2-minute speech here?
Trying to find back my screen. There we are. So I'm also trying to pull up my presentation. So first of all, thank you, Christiaan and Geert, for your very interesting presentations. I find it very interesting that we found a biological application and a non-biological application. So this was very, very useful. And also thank you for addressing the benefits for using a camera-based system. I was wondering, Geert, do you already have some results with our system, or is it still preliminary?
Yes, we do have results. Actually, also pH results. It is and also membrane fluidity results, and also snare protein. So we are having a palmitoylation mutant, and we could show that this misbehaves in different compartments. However, this is still preliminary, and it does not yet take full benefit of the kinetic resolution. So we're still working on that. We have some issues with the valve system. So if you add something, the sample moves. So it's a trivial problem, and I'm sure we can solve it. So yes, but we're using it on an almost daily basis. Yeah.
Good.
Remind everybody, if they have a question, please pose it in the chat.
Yeah, because personally, I haven't seen any questions coming by. Do you, Debbie?
I'm not yet. We have a quiet group today.
So am I now sharing my screen?
Yep.
Excellent. So I wanted to quickly go across the latest system because Christiaan, thank you for addressing the basics of the toggle system. As you mentioned, unfortunately, that system has become obsolete, and we move further with the VTOW system, which is currently being daily used by Geert van Bogaart. So it's a camera-based FLIM system, and well, everybody understands what FLIM means. It's a SPAD camera, so that stands for single photon avalanche diode, and what it enables us is to do very fast imaging, and that we can then utilize to do even faster frequency domain FLIM. It's a compact system. It basically has the same footprint as the toggle, and it uses only a single USB connection. Where the toggle was using camera link, the VTOW is only using a USB 3 connection, and it makes it a very versatile system. You can put it on any kind of microscope, and even your microscope can be in any kind of orientation, as Christiaan was showing. That was also for us very nice to see, and that it can be combined with all these imaging modalities as a spinning disk or TIRF spectrally, or you could also consider of thinking it maybe on a light sheet or just wide field. The specs of the VTOW system, it's 512 by 512 pixels, and the pixel size is just over 60 microns. Therefore, it has a field of view of more than 8 by 8 millimeters. Since it's pad, it doesn't have any read noise. It does still have some background noise, but it doesn't have any read noise anymore, allowing it to go to even lower light intensities. It has a peak detection efficiency of 50%. The sensor is equipped with micro lenses to increase the quantum efficiency, but we talk about peak detection efficiency. It doesn't necessarily means that you also have then like 50% QE. That depends on your setup. And typically it's in the order of 35%, comparable to our previous FLIM systems. And it can run up to 40 megahertz modulation, allowing you to do nanosecond lifetime measurements. The camera itself, it features some inputs and outputs for triggering your light source. That's the modulation out. We also have trigger inputs to make it suitable for automated screening applications. Requires water cooling to get it on a stable temperature. It doesn't have any deep cooling. It's just to enable for time-lapse measurements more stable performance. And it can also have additional output triggers to trigger, for instance, in a more high throughput setup. If you want to learn more, please feel free to reach out. It comes with our software, LIFA, and it enables you to do regular time-lapse measurements, single frequency measurements, and it gives you automatically the lifetimes. It can give you a lifetime overlay, instantly gives you the polar plot, and some histogram features. Also, it enables you to apply some filtering to get better statistical results. And we're working on our next release, and that will feature FLIM, so that you can also measure millisecond lifetimes. For the people who already have a VTOW system, that requires a firmware upgrade. So systems have to come back for a firmware upgrade, but then you can also do FLIM. We have some support for X and Y stage, so that you can do X and Y screening, and then they can be stitched together to get a bigger image. We have experiment control to do automatic FLIM recording. So instead of a whole slide, you could also think of maybe a 96 well plate that you can go back to your reference and then back to your sample. It has additional export features because it was already able to save, for instance, to TIFF, but this can be done automatically. Or if you already had done recordings in the past, that you can do a bulk export. This enables you to do easier analysis in your third-party software. And the polar plot will be more interactive. And there are improved trigger capabilities. And again, that will also require, besides a firmware, also a hardware upgrade to have these inputs and outputs. So if there are any questions about the technical hardware, please feel free to reach out.
Johan, just before we get into questions for yourself, we do have a question here for Geert from one of our attendees who has a hard stop on the hour. So if I could just pose this question for them. So Geert, the correlation of FRET versus reduction of lifetimes indicating interaction and the relative intensities of the donor and acceptor. You've had good negative results, kind of on-off situation. The data is very clear, but what if you see a moderate interaction and that those interactions vary with variation of concentration of donor and acceptor?
Yes, that's a really good question. Well, I think it's actually the advantage of FLIM and also the only way you can do this with fluorescence lifetime imaging, because it is absolutely quantitative. So for biologists, we all know that the FRET, the distance where you see first resonance energy transfer, depends on the distance between the donor acceptor to the power of 6. And for us biologists, that is by approximation a step function, meaning that you either have a condition of FRET or a condition of no FRET. So what you could do is you could actually make this absolutely quantitative, where you calibrate your system to the donor-only condition, where you have no FRET, and a condition where you expect 100% FRET. And then you can even say, if you can then get a subtle interaction, maybe 10% of your molecules interact, you will see a relative 10% reduction in the lifetime compared to those conditions. And you could even say, okay, 10% of my molecule is interacting. This is actually one of the things that we now try to do, but I think it's very sensitive. It's also, of course, dependent on expression levels, but only in the sense that at least in our condition, because we use primary human blood isolated cells, we still have the endogenous snare that is causing a background. So if you have higher snares, we get less interference from endogenous snares, and that is why we get variation in the data. I think if you do genomic tagging, you would lose that and you would get a really super sensitive system that is even quantitative, or you can make it quantitative. Does that answer the question?
Yeah. JW, does that answer? And we got it in time.
Yeah. We did have one more question come in. I understand that the Lambert system uses frequency domain over time domain. Can you explain the benefits? Johan, I think this one's posed towards you.
Well, the frequency domain allows us to go to a higher frame rate. And then in our case, it's also, we can stick then to nanosecond pulsed light sources. So there could also be an advantage there. For instance, for the wide field systems, this allows us to use LED excitation. So there's a lower requirement on the excitation light source, where typically in the time domain systems, you require very short pulsed lasers.
Okay, thank you. Any other questions? Please pose them in the chat. We have about 2 more minutes before the end of the webinar. And if there's any other questions, we'll get those. What I will do is we will end recording here, and this will be posted on our Lambert YouTube website. So I will be sure to follow up with all of you that have joined today, so that you can get the link to that. I want to send sincere thanks to all of our panelists today, taking time out of your busy schedules to talk with us about FLIM. It's been very much appreciated, and I hope this has been good for everybody in the audience, that you learned a little something. As Johan mentioned, if you do have any questions, we're here to answer those. Please feel free to reach out to us.